Hi, this is Natalie. And this summer, I worked alongside with Professor Barnabas Daru on this project that investigates the temporal turnover of microbial symbionts in seagrasses using herbarium specimens. Like their inland relatives, seagrasses also have those tiny microorganisms that resides within the intra and intercellular space of their plant cells. And in many plants, those microorganisms adopt a symbiotic relationship with their host, helping them with nutrient sequestration and fixation and establishing a stable biotic environment. However, the relationship between those microorganisms is poorly studied in seagrasses. And what we're trying to do here is to explore this problem by gathering the genetic information of fungal endophytes in Zostera marina. We choose this species because A, it is very accessible, it is native to North America, and is frequently found along the coast of California, known by the name of eelgrasses. And secondly, it is also a keystone species that provides countless ecosystem services to the coastal communities. As you can see on this graph here, it nurtures birds and fish and also helps improving the water quality. And however, unfortunately, the habitat of those species have been declining under the threat of various anthropogenic reasons, including climate change. And to understand how Zostera marina can better adapt to the climate may necessarily require a better understanding of their relationship with their endophytic microbes. Therefore, we wish to provide some helpful data that delineates how the composition of their endophytic community have changed in the past 100 years, and hopefully finding association with the changing mean annual temperature. What we did is to try extracting genetic information from archival herbarium specimens. As you can see here, which are those dry pressed plant samples. While they have been widely used in taxonomic researches, it remains an untapped repository for genetic information, especially of the microorganisms that lives closely with the plants. So we collected archival specimens from each of the 10 years intervals in the past century, and as well as from fresh samples from the Monterey Bay. Without damaging the overall integrity of the specimens, we took a small tissue from them for DNA extraction. And the result of the DNA extraction can be seen on this gel electrophoresis imaging. As you can see, some of the bands are barely visible, while those that are visible also tend to be kind of blurry, which indicates a compromised DNA quality. And by doing nanodrop reads on the extractions, we found this association between the DNA concentration and age, which is indicated by this multivariable linear regression model between the sample age, the nanodrop value, and the sample weight. What this model shows is essentially that with every 10 years increase of the sample age, the DNA concentration drops by approximately 0.37 nanograms per microliter. Luckily, this problem can be compensated by a two-step nested PCR approach, which isolates and amplifies the fungal ITSR DNA region. The gel images of those PCR amplifications are shown here and here. As you can see on this gel specifically, the PCR process gave rise to yet another difficulty. Since the DNA concentration of our template is really low, the forward and reverse primer may have accidentally complementarily a base pair and amplified each other, resulting in this short band at the bottom, known as primer dimers. And those primer dimers will interfere with the sequencing results. We tried to tackle this problem by experimenting with different concentrations of primers and found that 0.5 micromolar is able to yield the sufficient DNA amplicons without creating the primer dimer issues. So the next step of this project will be to prepare the library for all the PCR results and send the samples to be sequenced with next generation sequencing facilities. With the sequencing results, we'll be able to cluster different strains of DNAs into operational taxonomic units and study how the alpha diversity and dominant species of fungal endophytes could have changed over time. Eventually, we hope this information will help us develop better plans for predicting the changes in Zoster marina habitats and protecting the coastal ecosystem. We also hope that our research can bring awareness to the significance of herbariums, and which knowing that there's so much more to unravel in those precious archives.